In prophase one, the key events that we have are going to be synapsis, or the coming together of the homologous chromosomes. We're going to get crossing over between the non-sister chromatids that are within that tetrad. And then we're also going to get breakdown of the nuclear envelope. So those are the key events for prophase one, which is the longest of all of the meiosis phases. Once we pass through that phase, the next phase that we're going to go into is called Prometaphase One. So in Prometaphase One, the key event that we're going to get is going to do with the spindles. So we're going to get attachment, formation, and then also attachment of the spindles to the chromatids. And this is going to be really important because once we attach the spindles to the chromatids, then we're going to be able to take those chromatids and move them around inside the cell. So if we just sketch this out right here. So I have my tetrads, and then I'm going to have centrosomes located at both ends of the cell. And growing out from those centrosomes will be spindles, and those spindles are going to end up attaching to whatever tetrads I have inside the cell. So if I just kind of draw this here, we have some spindles that aren't attaching to those tetrads, but then we do have definitely ones that are attaching. So as soon as we get that good attachment of the spindles to the tetrads, then we're really ready to proceed into the next phase, because at this point we're really able to manipulate the position of the tetrads. The next phase that we get, and I'm just going to draw this one up here at the top, this next phase is going to be Metaphase One. And just like previous metaphases that we have looked at, this one does involve lining things up down the middle of the cell. And in this case, what we are actually lining up is going to be those tetrads which we've been moving around. So those homologous chromosomes, they are going to stick together for quite a while in that tetrad formation. And so if we just look at what we end up with, this would be the Metaphase One cell. It does look pretty unique and different from what we saw in mitosis, because in this particular stage, we actually have tetrads that are lined up down the middle of the cell. And we don't see that in the metaphase that we had in mitosis, and we're not going to see that in our later Metaphase 2 either. So as we look at these three phases that I've got listed here, these are the only three phases of meiosis when you're going to see the presence of the tetrads. Because as we move into the very next phase, those tetrads are going to separate and they're going to come apart. So our very next phase is going to be Anaphase One. In Anaphase One, the tetrads are going to separate from each other. We're also going to start to see an elongated cell, and that's because the cytokinesis process is beginning. And if we just draw what we're going to have inside this cell, when we do have the pulling apart of the tetrads, what we will actually end up seeing is that we will have entire chromosomes, and by entire chromosomes, really I'm talking about duplicated chromosomes. We're going to have whole duplicated chromosomes that are getting pulled apart and towards the two sides of the cell. Okay, so this is a look at that. So in other words, X's are moving towards the sides of the cell. So here we have the tetrads split. And when they do that, the homologous chromosomes separate. Now, it's not really shown in this picture as I've drawn it, but at this stage in time, the crossing over has taken place. So we would expect that really, if we could see it, there would be some recombinant chromatids that were moving away from each other in this particular image. Anaphase One does look very different from anaphase of mitosis because you have replicated or duplicated chromosomes, whole X's, that are being pulled towards the two sides of the cell. Now, as we take this Anaphase One cell and we move it into telophase, we're going to get telophase one. And in telophase one, we're really just going to separate these two groups of chromosomes now, and we're going to have the cytokinesis process take place as well. So we expect to get two cells, and just as I've drawn it here, we've got primarily the two blue X's in one cell, and we've got the two red X's in the other. That's definitely just an artifact of the way that I've drawn it. We're not necessarily going to have that, and there would be some recombinant chromosomes there as well. And so independent assortment, which would apply to Metaphase One, is going to affect the actual colors of the chromosomes that we get in these telophase 1 cells. So in telophase, we are going to get the chromosomes kind of reconfigured into two nuclei, which are going to be in those separate cells because we did have cytokinesis take place. So cytokinesis is what actually split the two cells from each other. But in addition to that, so we're going to rebuild the nucleus. And we will probably do some unwinding of the DNA as well. So that is the end of telophase 1. Now, I do want to point out here that even at this point in time, if we were to look at just one of these two cells that we got, these cells at this point, which are really just halfway done, these daughter cells, halfway daughter cells, they are haploid already. So they're haploid cells that are duplicated. And I say duplicated because that's the status of the chromosomes that are there, because they look like X's. But we do call it a haploid cell because notice that there's only one large chromosome in there and there's only one small chromosome, regardless of the fact that they are duplicated chromosomes. We also started with a cell that had 4 chromosomes in it, and now we have cells that only have 2 chromosomes in it. We're not done though, because we are going to take this cell and it's going, each one of these cells, and they are going to proceed into another cell division, which is going to be meiosis 2.